We're live again. Hello and welcome back to this week's Oxford Spark Science at Home live Q&A. We were having a little bit of technical difficulty there. We totally styled it out. Hopefully you're back with us. So yeah, we were talking drug discovery, dementia. What is dementia? What is Parkinson's disease? Remember, if you have questions, you can put those in the chat box. We'll try and get to those. Let's try this again, shall we?
Yes.
So hello again to Dr. Pavandeep Rai from the Department of Anatomy, Physiology, and Genetics, working on drug discovery and dementia. We were talking about dementia, how it's a collection of different diseases which have sort of brain cell death as a kind of common element to it. And we were just talking about the need to understand the basis of those diseases and how they work and the mechanisms that go awry to develop therapies and find drugs. So I suppose our next question should be, what are the existing therapies, and yeah, what are the hopes for future therapies?
Yeah. To start off with, the area of drug discovery in dementia has been really, really busy
Mm.
for a very long time. So lots of people have been making a lot of different types of drugs and compounds. And as I said before, all of these mechanisms that may be involved, these different therapies target a lot of those. But unfortunately, a lot of them are not actually able to cure the disease. And by cure, we mean taking the underlying biological process, making changes within the cell, and therefore correcting that disease. What we currently do have available for patients is something to treat more of the symptoms. And the way that these work are, we have two neurons and they send chemical messages between one neuron and the other. So when you have these neurons that are dying, for example, it may mean that the first neuron is releasing these chemical messages, but there's not another neuron around to receive it. So what these drugs actually do is they make that chemical message hang around for a bit longer. So multiple firings of that neuron may produce more chemical messengers and increase the probability of that message then being passed on to another neuron potentially in the area and improve the brain function or allow that brain function to be maintained. But obviously, with the progression of this, and it is a neurodegenerative disorder, so it does progress, when you have increased cell death, there's fewer and fewer neurons to catch that chemical signal on the other end. And therefore, you get to a point where these drugs are no longer effective within a lot of patients in advanced stages of dementia. So, one of the big areas at the moment is trying to really go back and understand the disease better. And the hope is that we can now start using genetic manipulation and gene editing techniques to really understand how these genes are affecting the outcome of disease. And why this is so important is because we have this idea, I think, that, oh, there's this one gene, it makes this one protein, and this one protein causes the problem within our cell. But actually, what we think now is that there's probably lots of different genes that are all interacting with each other, and they're causing a whole host of problems together. And that's what's then causing the disease. And by using genetics, we can go all the way back, right to the start, looking at the DNA and really understanding what are the relationships between those genes in the first place. Yeah, so it's a little bit there's kind of two sides to it. As you say, we can take patients and we can sequence their DNA, and then we can go back and have a look in their DNA what proteins, what genes, sorry, are producing mutated proteins, and causing or potentially causing the disease, say in Parkinson's patients. And on the other side of that, we can also do gene editing in more of a discovery role. So we take the entire genome and we do something called a CRISPR knockout. So we use a really, really specific gene editing tool, and that targets individual genes within our genome, and we knock them out individually. And now with the level of data that we can process and advanced computing techniques, we can actually look to see whether that specific gene has an effect on our outcome. And that means that really you're exploring the whole genome. You're not just limiting yourself to what might be present in patients, because that only gives you a snapshot of what actually could be affected. So that's kind of there's one where you go back from patients, and then there's one where you start right at the start, and then you test that gene knockout in, say, a cell model to see, does it have an effect that's similar to what we see in Parkinson's disease. Yeah, so we have this, as I was talking about, CRISPR-Cas technology, and that basically produces small entities that are able to bind to your gene of interest and cause that gene of interest to become mutated. So there's 20,000, generally we say that there's about 20,000 genes in the genome that we're interested in, and we can put all 20,000 of these chemical entities that target each of these genes onto our cells. And then what we do is we let them do their work, we let them knock out that gene of interest, and then we use an experiment to look at whether there are changes in the biology. So I was saying that these changes in biology can affect protein, they can affect pathways. So what we do is we've identified in our lab a couple of different pathways that we're interested in, and we can put this process through, we can run these cells in these specific experiments. And if we see huge shifts in those experiments, then we say, okay, that one is causing a symptom really similar to Parkinson's disease, what we see in patients, what we might see in animals. And then we can go and have a look at that gene, and we can use computational techniques to say, what else is that gene interacting with? What pathways involved in? Does it make sense? If it does make sense, then we can look at what do we already know about that pathway and put some context around it. Or alternatively, it might be a brand new pathway. And, you know, that's where the discovery and the excitement within science comes in. Yeah. So do you mean within, say, my specific lab, or do you mean, okay, yeah. So yeah, so as I said, it's very populated, and that's a great thing because we want as many options as possible. We want as many approaches really as possible because that increases the chances of finding something that may actually work. But for me specifically, I'm at that very early exploration stage of looking at what these genes are doing and what interactions are occurring. And the way that I hope that this would work is we would use these gene editing techniques and identify specific genes and really understand what they're doing. What are they doing within our cell models? So we have neurons that are made from stem cells that come from patients. So these patients will donate some of their skin cells. They'll get reprogrammed into stem cells, and then they're used to produce neurons within our lab. So they're very, very relevant cell models. And then we use those as cell models for the disease to say, okay, when we look at this gene, it has this effect in this disease model. And then what we hope is that we can then develop drugs to actually target the end product of that gene, so the protein that that gene might be making, and have a positive effect on the biology. The yeah, so you can, and you can actually produce very specific neurons. So you can say, I look at Parkinson's, I want to make a dopaminergic neuron, and you use different growth factors, chemical factors, and that reprograms, we call it, the cell into that specific neuron. But if you work in a different area, you can produce neurons that are affected in, say, Alzheimer's disease as well. But it is really cool. I've previously worked on muscle stem cells as well, and cardiac, so heart stem cells, and they even have spontaneous beating. So they're very, very highly relevant cell models. Mhm. Yeah, so the area of gene editing has really taken off in the past couple of years, and that has increased our understanding massively. We're now also able to sequence the genome much more rapidly, and we can do it for cheaper, so we can run a lot more experiments as well. So this area of really looking deep diving into genetics is really taking off and increasing our understanding. In terms of how close we are, the brain is very complicated. Uh, and with biology, as soon as you think you've answered one question, you've opened a can of worms to 10 other questions. So it's still very much. I mean, our knowledge is increasing hugely, but I think there's still a way to go to really interpret that knowledge. And yeah, that's why we need more scientists in that area, really, to look at the interpretation. But just quickly to talk about the cell models. So the cell models that we use in drug discovery, so very basic drug discovery, we use very simple cell models. But then when we want to do something that's very relevant to PD, we'll use very specific cell models. And then we also have some animal models as well that we can use. But really, the acid test is going into patients in trials. And that's where we really understand what's going on, and whether it's improving. So it's that level of complexity. So as you go along the process, your level of complexity of your organism increases until you get to a human. Mm. I mean, that can take a minimum of 10 years. It can take 10, 15, 20 years in total. It's a really, really long process. And what you hope is that you start off with lots of candidates. And then as you move across, as you go from your cell models, you go into your animal models, then you go into your patients, you reduce those number of candidates until you have one drug left at the end. And it's also important to think about when a drug is produced, it's not that it just is given to patients. There's continuous monitoring that has to go on. And sometimes the drugs have to be reevaluated, or they might be repurposed for slightly different areas. So, what's great about something like dementia is you may start off looking at a mechanism that's very specific for Parkinson's disease or Alzheimer's disease, but you may produce something which is actually still very relevant to a lot of other diseases. And so you may take a drug all the way through the process and then repurpose it for a slightly different disease area as well. But yes, a long process. No guarantee at the end that it will work, but you do all those tests along the way to really make sure that it'll have the desired effect. Mhm. I think for me it was I think specifically with science, it's the unknown, and it's that area of working in something where you aren't 100% certain. And the I think I liked the level of complexity and the questioning. And it's quite creative in many ways. It's a way of interpreting the world and making sense of the world around you, and that's what I really liked. And I think with biology, I was just fascinated by the fact that you can have, say, a piece of DNA which is a chemical, and it can go through all of these different areas and processes and make an entire organism. I think that really fascinated me, and I wanted to learn more about that. Really. Hmm. Yes. Yeah, so I went to the University of Birmingham and I studied biochemistry there. And while I was at Birmingham, I actually took a year out and I worked at AstraZeneca. So I did a year as an industrial placement student. And that's where I really got the bug for working in drug discovery and, working in an area that had quite a lot of impact in people's everyday lives. And when I was there, I was working on a specific project to do with mitochondria, which produce energy within cells. And they're implicated in a lot of different diseases. So I actually went to Newcastle after that and I did a master's and a PhD. And I did that in mitochondrial disease, so a very, very specific area, but really understanding the biology of these organelles. And I did that with Novartis, so another pharmaceutical company. And I actually managed to go and work out in the labs in Novartis as well. And the compound that we actually isolated from that is still under development with Novartis and with my lab, which was really nice. And then after that, I kind of wanted to move away from such a very specific biological process and apply it to something that has high impact and is more broad. So that's why I moved into dementia and came to Oxford, still looking in discovery, but now using an exciting different gene editing technique in order to do that work, but within Parkinson's. Um, Yeah, no. So we're currently collaborating with Bristol Myers Squibb. So everything I've done is generally with pharmaceutical companies, and they like to work with academics because we have a lot more freedom to explore these different areas and really ask the hard questions. And they can be focused more on the business, and we can do a lot of this much more discovery aspect of it. Mhm. Yeah, I think you're right. It's just a different setting, really. Mhm. And tapping into that different type of knowledge, I think, as well. I would say I would definitely say that if you're at university in your undergrad, get some lab experience. Being in the lab is very different to learning about being in the lab. So I would definitely get some experience, see if you like it. If you have the opportunity and you want to work in drug discovery, go away and work for one of the pharmaceutical companies. But just knock on people's doors in your university and say, "Can I work in your lab for a week or 2 weeks?" Everybody needs help. I know I have students who come and help me, and I really enjoy teaching them and working with them. I would say if you're in school as well, I know that my lab and many labs will actually have A-level students come and shadow us. Um, or go to science lectures, especially if you live in a city. They often have free science lectures. Feel free to go up to the scientist who's speaking at the end and just chat to them and say, "Hey, what's the best bit? What's the worst bit? What are you working on?" Yeah, just make as many connections and experiences as possible. And yeah, and enjoy school. Yeah, sure. So my group has a very active social media presence. They're Wade Martins. So if you Google Wade Martins group, Oxford, that should come up with all of the links. We have a great website that's got loads of videos. If you like robots, there's a lot of robotics on there. We use a lot of those to do our drug discovery work. So go and watch the videos. And my Twitter, I've actually written it down because I always forget, is PKRai191. So you can follow me. I'm on LinkedIn as well, under Pavandeep Rai. And yeah, feel free to drop me a message, and I'm happy to chat to anyone who wants to talk.